All right, so cell transport, this comes out of Chapter 7. And then cell cycle is going to be out of Chapter 10. I'm thinking we'll probably have a quiz on Chapter 7 before we move on to Chapter 10, just to make sure we understand cell transport stuff before we move on. But we'll have an end-of-unit test, just like we did for this last unit. So this is Unit 3, but we're covering 2 chapters. All right, first, a little bit about some microscopes. Okay, we'll be using a microscope in this unit, but there's only one kind of microscope that we actually own here at Vernon Hills. The other 2 are too expensive for us to have and take up like whole entire rooms. But the ones that we have that we'll be using are light microscopes. Now, you guys hopefully have experienced light microscopes from middle school, at least a little bit. Okay, a light microscope uses light to see the specimen. Okay, just like our eyes use light to see things. We talked about that actually when we talked about the Bradford assay and how light bounces off of objects. That's what gives us the perception of color. Like Diego's tie-dye t-shirt is bouncing all different kinds of colors back to your eyes, and that's how you see it. The thing with a microscope, though, is that not only does it use light, but it also uses a lens to magnify or make the object look bigger. That's why we can see really small things like cells with a microscope, but we really can't see them with our own eyes because our eyes don't have that type of magnification ability. A lot of times, though, in a cell, the parts that we're looking at don't have any color to them. Like the nucleus usually has no color to it. And so we have to use a stain, like iodine, for example, to see actual structures inside of cells. So we use stains and different colored dyes to kind of make certain parts of the cell stand out so we can see them underneath the microscope. And we'll be doing a lot of work with this. In fact, we're going to have a lab where we're going to look at your own cheek cells underneath the microscope. Remember we collected the DNA from them? Well, now you're going to see where that DNA came from because we're going to see the nucleus of your own cheek cells in a lab that we're going to do. So that's the scope that we use in class. And we'll talk about how to use that later when we need to actually use it. The other types of microscopes that we don't own here at Vernon Hills are electron microscopes. Main reason, these cost like millions of dollars.
Each?
Yeah. Thanks, Fernando. But you know what? We really wouldn't have much use. Well, it'd be kind of cool to have, but I don't know if we'd have much use for it here. You find these microscopes obviously in like research facilities at like big universities or even big hospitals. You don't really find these in high schools because they're really expensive. But just like a light microscope uses light to see an object, what do you think an electron microscope uses to see the specimens?
Wait, produces 3D images?
Yeah.
Sun?
Nope, not the sun. Jonas? Electrons. So instead of firing a beam of light at the object, we fire a beam of electrons. You guys hopefully learned in middle school, electrons are part of an atom. You have the nucleus in the middle, protons and neutrons sit in the nucleus, electrons are on the outside. Well, scientists have figured out that if we can bounce electrons off a specimen, we can use sensors to get that to capture those electrons and display an image. Okay, it uses a computer and some really technical instruments. But basically, it's electrons bouncing off the specimen or passing through the specimen, and then the computer kind of takes those electrons and forms an image out of it. Okay, it's a lot like if you think of how a bat. You guys learn how bats like navigate?
Animal.
They're blind. They don't see very well. So what do they use? Sound. They bounce sound waves off of like the walls of the cave and stuff, and that tells them where the wall is. Same idea. We just bounce little tiny electrons off of something, and then they bounce back, and we use that to make an image. Okay, but what's best about these electron microscopes is that they're used to see things that are incredibly small as 1/billionth of a meter. Okay, 1/billionth is 1 times 10 to the negative 9th meters. So that's a 1 with, so if I put a decimal point, that'd be 1, 2, 3, 4, 5, 6, 7, 8. That's how small we can see something. Incredibly tiny.
What's that little thing?
That's an M for meter. Sorry.
Looks like an O.
Wait, Mr.
You guys are so critical about how I write on this iPad. Yes, Carlos.
About like 1 millionth of a meter, is that like the maximum? I mean the minimum, like we can see?
We can't see that with our own eyes. With an electron microscope, that's what an electron microscope is used for.
How big is like a typical pencil?
I don't know. Good question. Maybe we'll talk about that later, though. We got to keep going, okay? All right. 2 types of electron microscopes, and they're abbreviated SEM and TEM. SEM stands for Scanning Electron Microscope. This bounces the electrons off the surface of the specimen. It gives us nice 3D images of the surface, but we don't get to see anything inside. So all we see is the outside part of the specimen. So if we're looking at a cell, this is great for seeing things like the cell membrane. It tells us the shape of the cell. We can see what's on top of the cell and what the cell membrane looks like. But do you think this would be good at seeing like the nucleus inside the cell?
No.
No, because we can't peer inside the cell for that.
How
For that type of...
How can we do it?
We're going to get to that, Carlos. Don't worry. Okay? For that type of observation, we need a transmission electron microscope, TEM. This, you actually have to take very thin slices of the specimen, which is pretty hard to do when you got really, really small specimens. You got to get really thin slices of it. But with that thin slice, we then shoot the electrons through the specimen. They actually go through it. They don't bounce off of it. But that gives us an image of the interior of what the specimen looks like on the inside. Now, let's think about this. If we're taking very thin slices of the specimen to look at, do you think that specimen is going to be still alive?
No.
No. Like if we want to look at the inside of a yeast cell, well, we have to kill the yeast cell in the process of making the slides to look at it, and so we're not looking at the organism alive. So that's the drawback. We really can't see the living and the functioning of it inside. We just basically see, what would look like inside, but the specimen's dead. It's not alive anymore. So there are some drawbacks to these microscopes. We can't really see living things with them. So here's some pictures of what different cells, these are yeast cells, would look like. Here's a light microscope, so we would see it like this. Now this has been colorized by a computer to give you a better picture of it, but they look like little tiny circles. Really can't tell much because yeast cells are very, very small. Here's what a transmission electron microscope would look like. Notice we see the inside. Okay, these are different colored organelles, like this might be a nucleus, this might be, I don't know, a vacuole or something. It's really hard to tell based on the image. And this would be a scanning electron microscope image. We see the surface of it. We can't see inside, though, but it gives us an idea of the shape of that cell. So, here's kind of how we decide what kind of microscope to use. If it's something really big, like a chicken egg, which actually is a cell, by the way, okay, we don't really need a microscope because we can see things that are 5 centimeters big, roughly. Yeah, Carla.
How cold is the virus?
How cold? It's a cold virus. It's a virus that causes you to have a cold, not actually a temperature. So, even some big unicellular organisms like this is a type of amoeba. It's about 1 millimeter big. We can still kind of see that, but not very much detail. So it's probably better to use a light microscope to see more detail of that. Your typical cell, light microscopes work fine. Even small bacteria cells, a light microscope works okay, but they're really, really tiny. So actually electron microscopes give us a better picture of those bacteria. Things like viruses or molecules like DNA, you definitely need electron microscopes to see those. A light microscope you won't be able to use because they're too small. Yes, Jeff? Just like a human egg is considered a cell. There's a lot of other stuff in a chicken egg. A lot of that chicken egg is just nutrients for the developing chicken. Because if you think about it, once the egg is fertilized, since it has that shell around it, the mother can't supply it with any nutrients. Whereas in a human, once the egg is fertilized, the mother supplies it with nutrients till the baby's born. Chicken egg, it relies on all the stuff that's packed in that egg. That has to sustain that living organism until the egg hatches. So that's why a chicken egg is big, because most of it is just nutrients inside there. Jonas?
large eggs? Is it like
Well, they have selectively bred chickens to produce bigger eggs. They basically say, oh, look, that chicken, this is getting into evolution a little bit. That chicken produces bigger eggs than this chicken. So chicken A produces bigger eggs than chicken B. Well, we'll let chicken A have more babies, and hopefully those babies will produce big eggs as well. And so farmers have been selecting which chickens to breed to produce bigger eggs because people want more for their money. It's done with a lot of different, cows are the same way. Cows are, the original cow, which came from an oxen, wasn't a big, fat, animal. It was actually pretty lean, but we have selectively bred them to suit our own wants and needs. Same thing with eggs, though. Yes, Carlos.
About the eggs, is it possible for the chicken in the egg to die?
Yeah.
Like before
it hatches? Yeah. But the eggs that you eat, though, guys, this is a big misconception. Might as well clear it up now. Are not fertilized chicken eggs. They would never have been chicks. Okay, so the eggs that you eat were not fertilized. They're just like a woman releases an egg every 28 days. Chickens lay an egg. I can't know how often, but they lay an egg, whether it's fertilized or not, every so often. Okay, it's just their eggs have a shell around them and actually come out, whereas women's are a lot smaller and don't. Anyways, moving on. All right, basic parts of a cell. So that's stuff on some microscope information. Basic parts of a cell, though. We've already talked about one. We talked about the control center. What was that organelle that contained the DNA? That was our control center. Who remembers? Nucleus. Okay, so that's already been talked about. This unit, we're focusing on the plasma membrane, sometimes referred to as the cell membrane. And we're talking about the cytoplasm, which a lot of people think is just the liquid part inside the cell. That's a big mistake. It's not just the liquid part. It's basically everything else.
Why does it say everything?
All right. So the cytoplasm is everything that's inside the cell membrane and outside the nucleus. It's just everything else in the cell. The liquid part has a special name called the cytosol. We're not going to really get into that too much, though, right now. So we're going to focus again on this plasma membrane.
I wonder why it's called plasma.
We'll talk about that eventually. Before we get into the cell membrane or the plasma membrane, again, I want to get you guys to get used to hearing that word because they're used interchangeably. Cell membrane, plasma membrane, basically mean the same thing. We got to talk about lipids because lipids are what make up the majority of what the cell membrane is made out of. So lipids are very large molecules, and we're not going to worry about this nonpolar part. You can cross that out. Okay? If you really are interested, it means no charge. So it has no positive or negative charge to it. But that's not that important for our understanding right now. But they are organic. So what does that mean? We talked about this first unit. We had trouble with the question on the test. Maybe we learned from that. What does organic mean, Jeff?
That's made of carbon.
Has mostly carbon.
And it's used DA or
Okay? So made of mostly carbon. So we should expect, when we look at a lipid, to have lots and lots of carbon atoms found in it. Really important, though, is to know that lipids do not dissolve or mix with water. Maybe you've tried this at home before, or done this in a middle school or elementary school experiment where you tried mixing oil with water, and you realize that they don't mix. Maybe you've had Italian dressing before. They like Italian dressing on their salads. Okay, and you have to really shake it up before you pour it out. So if you don't, the oil will settle to the top and the vinegar will settle to the bottom, and you won't get much of a good tasting dressing. Because oil and water don't mix. Oil is a lipid. Lipids and water don't mix. They don't dissolve. All right, building blocks. of lipids. 2 building blocks. Now, don't write all this down. Okay, I want you to write down glycerol. I want you to write down fatty acids, but we're not going to worry about the functional groups. And here's what I want you to know about glycerol. This is what glycerol looks like. Don't really need you to copy what this picture looks like down, but what I want you to realize is that glycerol has 3 places that it can form a bond to something else. I like to think of it as like 3 parking spots. So we can fit 3 other molecules, 3 other parts, 3 other building blocks to this glycerol. So it can hold 3 other things. And that's actually important for us to understand right now. The other building block are called fatty acids. In fact, this is where they get their name, the common name, fats from. Oftentimes we call lipids fats because of these fatty acids. They're long chains of carbon. So here's carbon, 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 carbon. Okay, all down this is showing carbon all the way down. It's a long chain of carbon bonded to each other to form this long tail-like looking, string-like looking molecule. It's like a long thread. So glycerol has 3 parking spots. Fatty acids are very long chains of carbon. So when we put them together, we get 2 different types of lipids. The first type we're going to talk about are called triglycerides. Here's our glycerol. This guy right here. 3 parking spots. 1, 2, 3. Here are the fatty acids. 1, 2, 3. Why do you think they call it a triglyceride? 3 what? And those 3 parking spots are filled with fatty acids. Okay? This is your typical fat molecule. When foods that you eat that are high in fat have a lot of these. Have a lot of triglycerides in them. When your body stores extra energy as fat, it stores it as this. Your cells actually store this in them for like a rainy day in case you don't have any food to eat. They can break down these fatty acids for energy. So one of the main functions of lipids is to store extra energy for a rainy day when you're starving and have nothing to eat. It's actually an evolutionary adaptation we have that we store fats so we could survive in case we didn't catch any food that day. But that has nothing to do with really the cell membrane right now, so we're not going to focus too much on that. The other type of fat, or lipid, are these phospholipids. And I do want you to draw this picture right here. I'm sorry, what? That's right, Juan. We did see that yesterday. There is a drawing feature to the slides you can use. You just got to search for it. But yes, I want to point out, we did see these yesterday. We saw this very same shape in that little activity we did. So let's kind of think back to yesterday. We talked about that this molecule called a phospholipid is 2 fatty acid tails. That's right here. 1, 2. And then it's attached to glycerol. That's right here. Don't worry about that. And then a phosphate, which is up here. That's the phosphate head. Now, thinking back to yesterday, part of this molecule was attracted to water, and part of this molecule repelled or had to be away from water. Who remembers which part was which? Which part liked or was by the water, was by the black part of your lab table, and which part had to be isolated? Peter?
Well, like the tail of things couldn't be by water.
Right. So these were the ones that the fatty acids did not like water. So I'm actually going to say they fear water.
Oh, yeah.
And then what about the phosphate head part, Peter?
It likes water.
It likes water.
It's fearless.
So we have actually scientific words to describe that. The first scientific word is hydrophilic. What do you think hydro refers to?
Water.
Water. Philic then must refer to, not Philadelphia, no.
Silly.
Nope, not silly either. Like if you,
Billy.
what's the only other word that's up here, guys?
Oh.
Likes. Likes water. Okay? So, you've never heard the word philic before?
Likes.
You ever heard on the news about, people or police officers arresting pedophiles?
Yeah.
What do you think that refers to?
Someone they like to.
Someone who really likes children. Pediatrics, the prefix ped refers to a child, and the ending philes comes from philic, which means to like or love. Now you know where that word came from. That's why they call them pedophiles, because they really like children a little too much, and so much they often get arrested for it.
Hey, don't
Okay?
do that.
That's what that word means. So let's use this though. If hydrophilic means likes water, how can we write a word that means fears water? What should we start it with? Hydro. Anybody know a word that means you're afraid of something?
It's better to fear kids.
Phobia. Hydrophobic is what we use to describe the tails that fear water, that push away water, repel water. They don't want to be in the water. They don't like the water. If you don't like going swimming, you might be hydrophobic too.
I like swimming.
My wife, she's what's the one where you're afraid of spiders?
Oh,
Arachnophobia. Yeah, she's a big fear of spiders. Arachnophobia. Okay? I guess if you liked spiders, you'd be arachnophilic. But that'd be weird. All right. Keep that in your heads, though. This hydrophobic, hydrophilic. We're going to use that when we talk about a cell membrane. So, a little bit about the cell membrane. It's the outer boundary of the cell. That's the easiest way of describing it. It's just the outermost part of the cell. It's what covers the cell's surface. It acts as the barrier. It's the container that the cell sits in. It's like it's an analogy to your skin. The outermost part of you is your skin. That's like the cell membrane. Okay? All organisms, all living things, all cells have a cell or a plasma membrane. That's one thing all cells have in common. Our cells have them, plant cells have them, Chewy's cells have them, even bacteria cells. The most simple cell on Earth has cell membranes.
That was
Okay, all cells have them. So, cell membranes consist of what we call a phospholipid bilayer. We looked at this yesterday. A lot of you got to this point where you... Yes, Samantha? These are online. Have you gone to the website? Lecture Notes and Documents? You go into Lecture Notes and Documents? That's where they are. If you want, I can show you after class. All right, we got to keep moving, though. So, we figured this out yesterday. You guys actually came up with this on your own. A lot of groups got this, which was awesome. That you figured out, well, since these phospholipids, the heads have to like water, and the tails have to fear water, have to be away from water, they have to face each other. The tails have to face the tails, and the heads point outwards. This creates this layer right in here, oops, that doesn't like water, fears water, and the outside likes water, and the inside likes water. This creates the boundary. This creates the barrier. We have water on this side and water on this side. But this is not just, a straight line. It's more of a circle. And actually, if it's a cell, it's more of a sphere. A complete covering of the cell, this layer. So all the tails are as far away from the water as they can be, whereas all the heads are next to the outside environment or the inside environment of the cell. And that's what creates that barrier. It's that phospholipid bilayer. Gee, I wonder why they call it a bilayer for. There's 2 layers of these phospholipids. They just point tail to tail. Okay, that's where we're going to end today. Okay, if you want to write that down, you can. It's what we already just talked about. But we're going to end there for today. We'll pick up here tomorrow.